Hello everyone, this is Himanshi Sharma. I've done my Masters in Zoology and I've qualified GATE Life Science exam twice in year 2021 and 2022. Today we are going to discuss about types of chromatin and C-value paradox. The topics we will be covering are genetic material organization, nucleosome, chromatin, role of H1 histone in forming chromatin, models of chromatin, types of chromatin, what is C-value paradox, and examples. Now the question is, how does approximately 2 meters of genetic material, that is DNA, in diploid cell get enclosed in 10 to 15 micrometer of nucleus? And the answer is, it is due to the chromatin organization. You can see here this is double helix DNA. Diameter is 2 nanometer, which gets wrapped around histone protein and forms nucleosome. Then the more compact form is solenoid, then supersolenoid is formed, and chromatid, and then chromosomes. Now the DNA is being distinguished in two types, one that wraps around the histone, which is core DNA, and one is linker DNA. So core DNA turns 1.65 times around histone octamer, and around 147 base pair length of DNA is being covered in this part. This is linker DNA, H1 histone protein. It is not being shown in this picture, but it binds at this position, and its length is around 20 to 60 base pair. And this whole structure looks like a beads on a string structure. These are the beads, and this is the string. And the whole diameter of this nucleosome structure is around 10 nanometer. Moving on to the next slide, you can see this is how the beads on a string structure looks like. This is nucleosome core particle. This is between these two structures is a linker DNA, and this is a nucleosome's electron micrograph. Now talking about the chromatin structure. So basically chromatin are nucleosomes that are arranged in a more compact form, that is higher order structure, and it is a complex of DNA, protein and RNA, and these are the values of DNA content 20 to 40%, protein content 50 to 65%, RNA content is 5 to 10%, and this structure is found in the interphase nucleus of the eukaryotic cells, that is the non-dividing stage of cell. And the function of this is to pack DNA into small volume to fit into the nucleus of cell and protect DNA in sequences. The diameter you can see is around 30 nanometer. See, you can see this is the nucleosome, 10 nanometer. In more compact form, it is taking form of chromatin. You can see here, these are the histone structures, H1 histone proteins. Now moving on to the next slide. What is the role of H1 histone in chromatin formation? That H1 histone we have read about is binds to the linker DNA. So H1 histone tightens the association of DNA with nucleosome. It increases protection from digestion by nucleases. These are some enzymes that degrade DNA. H1 produces more defined angle of DNA entry and exit from the nucleosome. H1 histone molecule bind on linker DNA, interact with each other, pulling nucleosome toward each other, thus are found responsible for packing of nucleosome in 10 then 30 nanometer diameter of chromatin structure. You can see it helps in the compaction of nucleosome into chromatin structure, and it protects the DNA from the nucleus damaged by nuclease, and it tightens the association. Now moving on to the next slide. Now here are the models for chromatin. Chromatin has two types of model. First is solenoid model and zigzag model. Diameter of both is 30 nanometer, but the difference is the solenoid one is regular and spiral configuration having 6 nucleosomes, 1, 2, 3, 4, 5, 6 nucleosomes per turn. Linker DNA does not pass through central axis. Here we can see linker DNA is not passing through the central axis, but here in this model, zigzag model, we are observing that the linker DNA is passing through the central axis and the configuration is not regular. The nucleosome configuration is not regular. Now moving on to the next slide. types of chromatin. We can see these are heterochromatin and euchromatin and heterochromatin. That this light one is the euchromatin, which is placed toward the center, and this dark one, which is more towards the periphery, is the heterochromatin. This is interphase nucleus. This is prometaphase nucleus. We can see in the prometaphase nucleus the more condensed form, that is chromosome is present, and in this interphase nucleus where we can see it is the less condensed form, that is euchromatin and heterochromatin are present. This is nuclear membrane and this is nucleolus. Now, as we have seen in the previous slide, that there are two types of chromatin, euchromatin and heterochromatin. Now we are differentiating them both, these both. Now, euchromatin is basically found in the nucleus inner part, and it is found both in eukaryotes and prokaryotes. And as it is thin, loosely packed, and lightly stained, that's why it does less amount of DNA in histone as compared to the heterochromatin, and it replicates in the earliest phase of the cell cycle. As it is less loosely packed, and that's why it is genetically and transcriptionally more active as compared to heterochromatin. And heterochromatin is found near the nucleus membrane, that is toward the periphery, and found in eukaryotes only. It is tightly bound and it is thick and darkly stained. It contains more amount of DNA and less amount, and that's why it replicates in the late as phase of the cell cycle. It is genetically and transcriptionally inactive, as it maintains the structural integrity of genome and regulates gene expression. Now we are discussing about the types of heterochromatin. So basically there are two types of heterochromatin, constitutive and facultative one. In constitutive heterochromatin, DNA is permanently inactive, that is, it doesn't participate in the cell cycle process. And whereas in facultative heterochromatin, the region of euchromatin gets converted sometimes in heterochromatin and at some point of development, and it can be either condensed or decondensed depending on the cell type. So constitutive one are more stable, and as compared to facultative, as facultative ones are reversible and can be converted into the euchromatin again. Constitutive heterochromatin are made up of satellite DNA, and these facultative one are made up of line sequences. Now the occurrence where the constitutive heterochromatin is present is at the telomere, that is the ends of the chromosome, at the centromere, around the centromere, in the C bands of chromosome, and on the secondary constriction bodies, that is satellite body. We will see these structures in the next slide. And facultative heterochromatin, it is basically what inactivates the X chromosome in the somatic cell of female, and it forms a bar body. Now these are the structure where this is the constitutive heterochromatin positions, telomere, centromere, and orange colored indicates the heterochromatin one areas, and this is the bar body, which is the inactive X chromosome formed due to the facultative heterochromatin. Moving, now we are discussing about C value. So what is C value? C value is basically the amount of DNA in haploid genome of an organism, and it is calculated in picograms or expressed in base pair. So earlier it was thought that according to the complexity of organism, the C value, that is the genomic size, increases, and it was related as a linear relationship. But later on this concept proved wrong, and the concept of C value paradox came across. Now, what is C value paradox? It was given by the C.A. Thomas in 1971, and it shows that the complexity of an organism does not show linear relationship with the genomic size of an organism. And the examples are in human beings. As we know, human beings are obviously more complex than the salamanders, that is amphibians. Still, the genome size of human is 3.2 billion base pair, and whereas in salamander the genome size is 120 billion base pair. That is, salamander have 40 times more DNA than humans. So, and another example is Musca domestica and Drosophila melanogaster belong to same order, complexity same. Still, the genome size varies significantly by around 6 times. That is how I have 8.6 into 10 to the power 8 base pair of genome material, and this Drosophila has 1.4 into 10 to the power 8 base pair, and it shows that housefly has 6 times more genome size than as compared to the Drosophila. Now here is a graph which is showing that the genome size of organism is not showing linear relationship with the complexity of organism, as we are seeing that this is obviously not linear. Mammals should have more genome size, but it is not, it doesn't have, and the linear relationship that was once thought that would be linear is not actually linear. Now moving on. So why this paradox came across? The genome content of higher organisms, that is like we mammals, also have non-coding repetitive DNA sequence called junk DNA. This leads to disturbance in the relationship between or complexity of organism and its genome size. So this is the end of our discussion. Thank you very much. Goodbye.